Yeah, so today I will talk about respondent-driven sampling and some potential sources of bias in this sampling process. So one challenge that researchers in public health and social sciences face every day is how to reach some kind of populations, because they are very difficult to reach. There are many reasons for that. That's because the members are rare, or perhaps they're stigmatized, or even illegal or unregulated somehow. So, for example, we have sex workers, MSM. I should say that this is men who have sex with men. I remember the reviewer didn't like this term, but that's the standard term in public health. You have illegal immigrants and regulated workers, so there's plenty of populations that are difficult to sample using the regular methods. And essentially in these populations, it's difficult to define a sampling frame. So one solution to sample members of these hidden populations is to navigate through their social networks and recruit people using snowball sampling. So what's the idea of snowball sampling? Essentially, you find a seed in this target population, and then you ask this seed to invite some friends or some other people that belong to the same community or population, actually. And then you go to these invited people and you ask them to invite other people. And this goes on until you reach the sample size that you like, or until no more people can be invited. So one problem of snowball sampling is that in social networks there is a lot of heterogeneity in the number of contacts. So some people have many more contacts than others. So it's more likely that they will be reached more often, or at least they are more easily reachable because they have high degree. So for this doesn't work. But for example, this guy in the middle, you see that there are many other people that can reach him. So the idea of respondent-driven sampling, which was a method developed by Hackathorn 20 years ago, is to combine this snowball sampling with some kind of weighting procedure to compensate for the fact that some people can be more easily invited. But then another problem of respondent-driven sampling, and actually also from snowball sampling, is that social networks are highly clustered. That means they have what we call in network science community structure, which is like groups of nodes that are very connected between themselves, but loosely connected with nodes from the other communities. So this is like a more random structure, and there you can see this kind of community structure. And one important thing is that when you have community structure, you have some nodes that act like bridges between these communities. So they are actually like bottlenecks where any diffusion process have to pass through them to reach another community. And this has quite a lot of consequences for this RDS sampling. I can refer to some other papers if you want to see other problems of RDS, but today I will focus on this one. So my research question in this paper was what is the impact of this community structure in respondent-driven sampling? So to address this question, we did a computational experiment. So basically we simulate a social network, and then we also simulate the sampling process. So first, we basically want a network with this strong community structure. So this is a picture from that paper in a physics journal. And then basically this is like a more realistic social network. You see there are like communities that are very small and some other communities that can be very big. You can think, for example, if you think on sex workers, one community could be like female sex workers, another community could be male sex workers, another one could be transvestites, and so on. So actually you see these social networks, this community structure in all kinds of social networks. So they are pretty ubiquitous. So what we did actually try two methods to build these artificial social networks. I was thinking that I wouldn't have time because I thought it was only 15 minutes. So I planned to talk only about one of the methods. And this one is actually an algorithm devised by Lantichinetti and Fortunato in 2009. They are physicists. I am too, so don't... I guess I'm the only physicist here, but anyways, that shouldn't be a problem. Yeah, so basically the idea of this algorithm is that you have a configuration model, so you connected links randomly, but they are constrained by some kind of community structure or predefined communities. So there are two things fixed in the model. You have the degree distribution that is fixed. It's a power law, because we like power laws in physics. And the community size distribution is also a power law. But you can choose the parameters of these power laws. And I chose two values that are close to real systems. And then you can also choose the network size. I chose like 10,000. You can choose the number of the communities and the range of the community size. But remember that each community will be sampled, the size of each community will be sampled from this distribution. And then you can also choose the number of overlapping nodes. So overlapping nodes are basically nodes that belongs to different communities. So they will be these bridging nodes that I mentioned before. And the mixing parameter is just to control how many connections you make inside and outside the community. So I will not go through how to build it, but you can, there is actually a software that you can borrow to do that. But essentially, it creates this kind of structures very nicely. And then another step is that we also simulate a prevalence of some feature or some trait in this social network. And then we tried three different methods. So the first one is just like a reference model. So we pick 25% of the nodes of this population, and then we infect them with some feature A, with some trait A, or with some disease, depends how you want to call it. And then the second method, we also pick 25% of these nodes, but then we first infect nodes in the small communities, and then we go to the next biggest one, and so on, until 25% is infected. And the same is for the third method, but then we first infected nodes in the largest communities. We also add some noise to that's to remove homophily. So homophily is this tendency, right, that people that are similar, they are connected. So for example, let's say people that are male, they have more male friends. That may not be true, but yeah, that's the idea. But anyways, we redistribute 40% of A just to remove a little bit of this homophily. So the infection is more spread in the network, although it's mostly either in the smallest or in the largest community. And then we simulate the respondent-driven sampling on these networks. And essentially, we start by selecting 10 seeds. There are 2 ways to select the seeds. I will tell you this later. And then each participant can invite 3 friends that were not invited before. And then with probability P, each of these friends can invite 3 other friends. And this goes on until we reach 500 individuals in the sample, or until no more people can be invited. And one important thing is that we do sampling without replacement. And the other things are not so important. But basically, we chose these parameters because that's what people more or less choose in real RDS studies. Yeah, so here there are like different estimators to calculate the prevalence of some feature using RDS. I chose this one, which is called RDS2, because this is the most popular one, at least among sociologists and people in public health. And there you can see how this weighting procedure works. So basically, it's divided by the degree of the node. But then for the study, we take the average and we calculate the bias for 500 realizations of the sampling process. This is actually, I should mention that there is like lots of estimators, and sometimes there is a healthy fight between people, like claiming that they have a better estimator than the other people. Yeah, so this is just like a reference result. So basically the network here has no communities, so it's a random network with power law degree distribution, but otherwise it's random. A is uniformly distributed, also 25% of the people are infected. And then we see that the estimator performs very well. It's very good, except when the response rates, or this probability P, is below 0.4. But that happens because the sample size is not very big, or actually it's kind of small if the response rates are small. And the bias, here I put in the percentage in respect to the 25%. So we also see that it can be quite large if the response rates are small. But then here it comes a more interesting thing, I think. So in this case, the seeds are chosen uniformly in the population. So those 10 seeds are just chosen uniformly. And then we have the infection concentrated in the small communities. So what we see here is when there is no noise, there is quite an underestimation of the prevalence, which is relatively high. But if you add some noise, this decreases. So of course, if you, oh yes, yeah, great. It depends very much on the strength of the community structure. There are some methods to calculate this strength, but here just show like a strong network community and what happens. So we see this underestimation, and also the bias can be quite high. On the other hand, if the infection is concentrated in larger communities, we have an overestimation of the prevalence. And these are also the standard deviation. So it's actually larger than in the previous case. So what happens here is that essentially if you think in the sampling process as a random process, like a random walker, it's not too difficult to understand why this happens. Because essentially like the random walker may get trapped inside these small communities, and then that's why you end up, oh yeah, sorry, I'm saying a different thing. I mean, it can get trapped, but it can also not penetrate in the smaller communities if you just start randomly. Because you have to remember that it's more likely, even if you choose uniformly, it's more likely that the seeds will start in the largest communities. So they will have problems to go into these smaller communities. And that's why we have the overestimation here. So here we did a slightly different experiment. So it's also like infection can be concentrated either in the small or in the large communities, but the seeds start in the small communities. So then you see that the prevalence is considerable overestimated when A is in the small communities. And on the other hand, it's underestimated if A is in the largest communities. And then, quite naturally, if the seeds start in the large communities, also we have an underestimation if A is in the small communities, but if A is in the large communities, we have an overestimation. So again, this is essentially the problem that the walker cannot like move so freely when you have the community structure. So if you think on these bridging nodes, essentially when one of them is sampled, you cannot go again through this route, either inside or outside the small communities. And then just to summarize, so social clustering, represented as community structure, is happens quite often in real systems. So it's very important structure, and that creates a lot of heterogeneity in the system, in the social system. And then the estimators don't really take this into account. So the estimators that we use are probably giving some biased results. Or at least, I don't know how much, but because we actually don't know normally the social structure or the social network. And then the prevalence may be under or overestimated depending on different factors, like where the infection or the feature is concentrated, or how you choose the seeds and things like that. So there are some recommendations. This is more like for people actually working with RDS, but it may be more interesting to restrict an RDS study to certain locations or to certain groups. So you don't have this problem that there will be very few people connecting different groups. And then we try to select seeds as uniformly as possible. This is very challenging. I work with some people. We actually are doing some real RDS studies, and it's very difficult to choose the people randomly, as we assume in theory. And also it's very difficult to control the response rates, but it's better to use at least, or aim to have at least 0.4. But they laugh at me when I said that. Yeah, so I want to thank you. This is where we published these results. It was published last year. But I also want to invite you to take a look into other papers related to sampling. The first one in arXiv, in particular, is also a new estimator for disconnected networks. And the second one is not about respondent-driven sampling, but it's about sampling in temporal networks. So thank you.
This is a joint work with my colleague Vincent Miel, who works in Lyon in France. So it's a paper about how to model networks observed through time. I will start with a brief introduction about what we are interested in, which are mostly contact networks. And first, with the static case. So, in the contact networks I want to look at, there are different individuals that may be at stake. I can be interested in humans or animals or even species, because I've been working a lot on applications to ecology. And these contact networks may be very diverse. They can be built from different type of measurements. So the top of data, let's say, top class data would be sensor-based measurements, where you have some chips that you put on the animals or even humans. Let's carry a chip on something like a device like that, and you follow the activity of this individual through space, and you can record interactions between two individuals. But you can also use like declarations in surveys. For instance, if someone tells you, okay, my friends are this one, and this one, this can also be used as data to construct this type of social networks. In ecology, you have also a lot of field observations where there's some people just observing the animals and writing down all the contacts he observes between each individuals. Or also you have species interaction like trophic relationships. When you know that this kind of species eats this kind of species in that area, then you record an interaction between those two species. And there are also trapping data when you capture some animals into specific locations, and when you capture two individuals at the same location within the same period, you consider that they may have interacted. So there are a lot of different data, and I want a model that fits all of this kind of data. There are different aims behind this work. One of the main aims here is to study sociability. This can be between humans or animals. And there are also ecology questions when I work on animals or species. And the formal definition is that I have n individuals that will be the nodes of my network, and I can record two different types of interaction, either just a binary interaction, so I record whether there's a presence or absence of contact between any two individuals, or I can record weighted interactions, like for instance frequency of contact between these two individuals. So the networks that I will look like can be either binary or weighted. And I want to look at dynamic contact networks. So here is a representation of what I mean by dynamic contact networks. Very basically, these are snapshots of contact networks registered at different time steps. So these time steps can be hours, days, weeks, or seasons, or anything else. And in the picture here, all the individuals, the nodes, are the same through the networks, but in practice, some individuals may be present or absent at each time step. In particular, when you work on animals, then through seasons, for instance, then you will have animals that disappear. So it's important that you account for some small variability into the presence or absence of the individuals. You cannot have completely different sets of individuals, otherwise it's not a longitudinal study, but some sort of variability is okay. So the formal definition of what I mean by dynamic contact network is that I have T, capital T time steps. For each small t, I have nt individuals, which are the species or the animals that are present at this time step. And in totals, I have capital N individuals, and this capital N, it's way smaller than the sum of all the NTs, meaning that I need some redundancy into the presence of the individuals. Okay, most of them must be always present. And then I record either a binary information, presence or absence of contacts between two individuals, or a weighted information, like the frequency of those contacts. So what do we want to study dynamic contact networks? The main question here is there a social structure? So in the previous talk, Luis talked a lot about the community structure. Here I'm interested in any structure. This can be communities, this can be anything else, okay? So I wanna understand if there's a peculiar non-random mixing between the individuals at stake, and that would be a sign for social organization. And here I looked at that in a dynamic setup. So if there is a social structure, I want to identify what is its dynamics. Other questions could be asked here, like for instance, what is the variation of this structure with other factors like the seasonal changes, the breeding seasons for animals, response to stress, and many other questions like that. And I could also look at specific questions like how can we predict infectious diseases spreading according to this inference of the social structure. In this specific talk, I will only just focus on the two first questions. Is there a social structure, and what is dynamics through time? And our answer here is that we want to move beyond descriptive statistics and propose a statistical model for the organization of these networks. And to do that, we want to rely on a clustering of the nodes to capture this social structure. So that's the main work that we've done here. So the one important thing that I want you to understand before you leave this room is what's the difficulty in clustering dynamic networks. If you cluster one network, let's say this first one, so you cluster the nodes, meaning you find some colors for the groups, and I'm not specific here about what the group means, okay? But I want to find groups, and I find those groups in my network in some way. Then you certainly know that the answer that you get is defined only up to a label switching of the groups, meaning that if these two colors, blue and yellow, were switched, then the result would be the same. So now, if I cluster this first network at time t1 and this second network at time t2, I have these two clusterings, and I cannot compare them, saying things like, okay, this node here, he has changed it from the blue group to the yellow group. Because I could have switched blue and yellow in the first network, and then I would have a completely different interpretation of the groups a long time, okay? So what's important here is that if you want to do a dynamic clustering of your network, you need to do that globally. You cannot do that time by time, because otherwise you cannot interpret what's going on with the groups a long time. So what I really want to do here is answer questions like who is changing group between two time points. And to do that, I need a smooth recovery of the clusters across time. So that's the main point of this work. So what we have done before entering into the details of the model, which won't be long, our contributions are the following. We have proposed a model that is a dynamic stochastic block model that I will explain in the next slide, and it enables to make groups that you can follow through time on your networks, on the nodes of your networks. So here, the graphs may be directed or undirected, the same for me. They can be binary or weighted, okay? You can have different kind of information on the interactions. And the groups will change through time, that's the goal, or they can change through time. They are not mandatory to change, but they can change through time. And we also allow the parameters to change through time. So the parameters of each model, they can be specific to the time point you're looking at. And so this is a bit tricky, and what we've done is that we have carefully discussed identifiability conditions on the parameters for this to be possible, okay? And then, once we have set up this dynamic stochastic block model, what we've done is to propose an inference method for this model. So what we rely on is a variational expectation maximization algorithm. So that will infer the groups across time and the model parameters. And we also have a model selection criterion, which is of the type of an ICL for integrated classification likelihood criterion. So it's only some sort of penalized log likelihood criterion, okay? And this criterion helps us to select for the number of groups, because when you want to cluster the graphs, sometimes you have no idea of how many groups you want. So you can select them with that criterion. So the model, I will explain it in a few slides. First, I need to explain what is the stochastic block model. For those who don't know about this model, it's very popular, but not everyone knows about it. So the stochastic block model in the binary case will help modeling the static networks. So the idea is that you observe this network without the colors on the nodes, and you assume that there are Q groups that will be represented by the colors, and you have latent random variables, which I denote here by ZIT. So this I is for the index of the node. There are n different nodes, and T is for the time point. So these are IID random variables that take values in the Q different groups, and they are not observed, okay? And what you observe is presence or absence of an edge at time t. We are in the binary case for now. And you assume that this is given through an adjacency matrix Yijt. So it's full of 0 or 1, telling you whether node I interacts with node j at time t. And in the stochastic block model, that's very simple. You assume that conditional on the latent groups that you do not observe, the random variables are independent, so the interactions are independent, and they follow just a Bernoulli distribution with a parameter beta that depends on the colors of the two nodes that you are trying to join. Okay? So that's very simple. Here, between this blue node and this green node, I don't have an interaction, and that's done with a Bernoulli distribution with probability some beta blue-green. That's the parameter. So this can be easily generalized to the weighted case. In the weighted case, you have the same thing. Latent variables are the same, and observations are weights. You either observe a true zero, namely no interactions between the two nodes, or you observe anything. Let's say a value in R, or even a vector valued. And then you put a parametric distribution. You model the true zeros with this Dirac distribution with some sparsity parameter beta. And the non-zeros values, you can put any distribution that you like the Gaussian distribution if these are real values. This can be a Poisson distribution if these are counting values, anything that you want. But it's a parametric model. With this at hand, what we've done is that we have combined this static stochastic block model with some dynamics to handle to produce the dynamic stochastic block model. So what we've done is that we have assumed that the latent variables that gave you the groups between the nodes Across individuals, the ZI are still IID, independent and identically distributed, but through time, each ZI is in fact a stationary Markov chain. So here, if I look at the first node, I have this stationary Markov chain through time that tells me how the group of this node will change through time. And at some specific time point t, if I know all the groups of all the nodes, then I have a static stochastic block model with parameter that depends on t, that gives me the adjacency matrix at time t from these latent groups. Okay? So that's the main idea of the model, plus there are some conditions on the parameter for it to be identifiable. I didn't put them here because I thought I would run out of time, and I think I'm right. So how long do I have? Okay. So that's all for the details. Okay, I want it to be light. And I'll show you some simulations and a real data set. So first, in the simulation, we tested the clustering performances of our model. So we looked at one index, which is the adjusted run index, and we looked at it on the world classification. So that's what I call the global IRI, meaning that we looked at how the node I is classified at all time points globally. And some people in the literature had looked at another criterion, which was an average adjusted run index that was computed at each time step, and they averaged it from all the time steps. And this was not rigorous because you don't account for label switching across time. So this criterion is not good, but we put it in order to make comparisons with other works. The simulation setup are the following. We looked at binary graph with 100 nodes and 2 different values of t, either 5 or 10 time points. There are 100 datasets for each scenario. There are 2 latent groups, and the probability, the transition probability for the Markov chain here takes 3 different values from low to high stability of the groups. In this case, you see that the groups change very little from one time point to the other one. In this one, the groups will change a little bit more. And we have also, let's say, 4 different scenarios for the connectivity parameters, from low minus easiness to medium plus easiness, meaning that here it's more easy to distinguish the groups than here. Here the values of interactions for the groups are 0.2, 0.1, 0.15. They are very close, so it's very difficult to distinguish the groups. And the results are the following ones. So here in the first row, you can see the results for t equals 5 time points. In the second row, you see the results for t equals 10 time points. From the left to the right, you can see from low group stability to high group stability, meaning that it's easier from the left to the right to infer the groups. And inside each box, you can see the different values of beta from low minus easiness to medium plus easiness. So it's easier from the left to the right. And what is depicted here is the box plot of the adjusted run index. So the adjusted run index is very good when it's equal to 1, saying that the classification that you've recovered, it's the true one from that you used to simulate the data. And so you can see that when the problem is difficult here and here and here, we cannot infer the groups properly. But when we go when we increase the easiness of the problem, then the box plot becomes equal to 1, and the clustering is perfect. Okay. We also tested the model selection, and we checked that in a setup with Q equals 4 groups. Most of the time, our criterion recovers exactly the 4 groups, And when it doesn't recover the 4 groups, in fact, it's because we had a very bad classification obtained with this parameter, meaning that we did not succeed in optimizing our likelihood criterion or something like that. I will finish by just showing you what we can obtain on a real data set. So this data set is a social network between humans. It was taken from the Social Patterns website, which is a great website with nice data to test your method. So here specifically, these are face-to-face encounters of high school students. So they had wearable sensors, a sensor that was put in something like that. And each time the students were face-to-face, then the sensor recorded a contact. The experiment lasted for 4 days, and there are 27 students in this experiment. So we have recorded the frequency of interactions, and we have discretized this signal into 3 different beams with medium, low, and high interaction levels, and we have selected 4 groups. And here you can see one of the outputs of our algorithm, which tells you, so in each column, these are the 4 different days, the 4 different values of the time. And here you can see the 4 different groups. And here you see the dynamics. This is some kind of flow plot of the individuals inside the group. So you can see that the group 4 at time T1, essentially they stay, all these individuals, they stay in group 4 at time T2. And they are joined by some individuals from the group 2 that join them from time T1 to time T2, okay, etc. So you can see the flow of the groups. So you see group 1 and 4 are very stable, and only group 2 and 3 have some exchanges with the other groups. You can also output the parameters of your stochastic block models. So this is a little bit tricky to interpret. I don't know if I have much time, but here the width of these plots tells you about the interactions between all the individuals, how much interactions are there. So if here in this case, for instance, it means that the network is very sparse. There are a lot of zeros, and here not many zeros. And then the height of the colors, these are the values for the 3 different beams: medium, low, and high interactions. So here you have high interactions which have the largest probability in a sparse environment. So you can interpret the groups with the characteristic of the groups with those parameters. So in the paper we have a lot of different description of these groups. Some are communities, other ones are not really communities, and they exchange students. And we have crossed this information a posteriori with the gender in order to interpret what's happening on this network. I'm not entering into details because I'm almost finished. So let me just conclude on this presentation. So what we've done in this work is to propose a dynamic stochastic block model. It enables you to reconstruct the evolution of groups on the nodes through time. And most importantly, it controls for the problem of labial switching issue between the different time steps, so that you can really follow the groups. You can have either binary or weighted dataset. They can be directed or undirected. It will work. And you can select the number of groups through an ICL criterion. There's an R package, which is called DIN-SBM, that's available on the CRAN. And the paper has been published in GRSSB this year, in 2015. And also we have written a companion paper for ecologists that has appeared in the Royal Society Open Science this year also, that is to help them understand what we do and encourage them to use our method. Thank you for your attention.
And again, this is a paper in the special issue of JSSC on networks, and the title is Estimating Whole Brain Dynamics Using Spectral Clustering. So this is a joint work with Ivor Cribben, who is an assistant professor in Alberta School of Business in Canada. So maybe I'll start this talk by answering or by commenting on one of the questions the previous speaker got asked about the evolution and the changes. So I don't know how you guys think about this question. In my understanding, maybe we can say these slow little changes, we call it evolution, and we can model it using some locally stationary ways. And if we have big changes, abrupt changes, then we may go for another way to say, okay, probably there's a change point detection problem, and we'll go for another way. Everything here depends on two things. One is how big this change is. This, of course, in different, if you have different methods or whatever, you have different way to answer this question. And also how separated those two consecutive different points are. So I guess everything is very subtle. And everything is method or theory dependent. But I just want to make a comment here. Here I do a change point detection. Okay. This is so the outline of today's talk. I'll talk about the application background, why I got interested in this question, and the two main technique components of my methods. The theory of this talk, which is in a separate paper and which I will present it in tomorrow's talk. Tomorrow, 11 something, right, Heather? Great. So far in these 25 minutes, there's no theory. Let's start with brain. The application background of this talk is about the brain imaging. Ivor has done a lot of stuff in the brain imaging, and he brought this problem to me. He said, okay, can we do something to the resting state fMRI data? So as for the fMRI data, this is a very powerful, widely used technique to study our brain. So what we actually observe, so for instance, we call it imaging, but it's different from just taking a picture. It's actually the measure of the BOLD. BOLD is the blood oxygen level dependent. So this is basically to say, if a certain area, if a certain region of your brain is activated, is more active than other parts, then obviously this active part needs more oxygen. And the blood carries the iron, and basically this BOLD is to capture what should I do? Yeah, so BOLD is a way to measure how active a certain region of your brain is. So for instance, I can't find it. Okay. So for instance, say if a subject is in an experiment and he or she is lying in this machine, fMRI machine, and this is along the whole time course. And what fMRI is doing is it partitions the brain into many, many little voxels. Usually one voxel is 3 millimeter by 3 millimeter by 3 millimeter. And each voxel gives you one BOLD measurement at one time point. As you can imagine, for every voxel during the whole time course, it gives you a univariate time series. Okay? This is where the time series kicks in. But if you think, okay, I just take one snapshot of my fMRI data, then there are roughly speaking, 60 by 60 by 60 voxels. So this is a high-dimensional network because our brain is working in a collaborative way. So that's where the networks kicks in. Okay, so this is the data, and this is part of my motivation. So in order to study the brain, There has been a lot of papers from many different areas studying the brain, developing methods, previously people are more interested in the localization to say, okay, if you ask me to conduct a certain task, which area of my brain is really associated to this task? So this is a localization problem. Recently, people are more interested in the functional connectivity. This is to study the collaborative way the brain is presenting. And there are many clinical papers to say, okay, we should do this. I just submit that part. I'm not really an expert there. Okay, speaking of this functional connectivity, this is really a very vague word. It's basically to say, okay, different parts are associated, are correlated. By saying these words as a statistician, of course, we can think of, well, we can use correlation matrices, covariance matrices, and if you are doing things in the frequency domain, you can probably think coherence matrices, partial coherence matrices. So yes, indeed, there are so many papers already been using all this kind of technique to study the brain connectivity, the functional connectivity. All sounds good. However, all these methods, all these well-established methods, they rely on very strong condition. This is to say, our brain time series is stationary. No one will buy that. So what can we do? Also, there's a very natural extension of all the previous methods we mentioned is to say, okay, without assuming that the whole time course, like if you lie in the fMRI machine for 1 hour, I can't assume this 1 hour to be stationary, but I can always assume that in a short segment of time it is stationary. And then using moving window, I do whatever covariance correlation coherence, partial coherence in a very small segment of time, and then move this window to get my results back. Yeah, this is good, but can we do something else? This moving window technique also has some limitations. How will you choose the bandwidth? The window width, or what kind of kernels? If you think this moving window is just a kernel, only assign 1 or 0 value to your time points, you can think I can use some other kernel. There are so many subtleties, so we go for another thing. We do change point detection. The idea of a change point detection that comes back to what I said at the very beginning of my talk, is to say, okay, this is a very simple toy example. So in the left panel, what you can see, the black curve is a realization. It's basically white noise, and the red line here is the underlying truth. That's the real signal. So if you just see the red curve, you can see I have 2 changes. These 2 big jumps, okay? And I get these realizations here. So what I want to do is, if I only have these black realizations, can I find the true change points in my underlying truth? Okay, so this is a very well studied technique. It's called Q-sum. It goes back to 1954. The idea is to say at every time point, okay, in order to test whether or not this is a potential change point, I split the data into 2. I split the data into the one previous to this certain time point, say 50, and all the time points after 50. For these 2 parts, I calculated the standardized sample mean, and then I just calculated it's actually balanced by the sample size. So and then I calculated this normalized sample mean difference, and that will give me a measurement which is called a Q-sum of a certain time point. Okay, so in the right panel, the black curve here is exactly what you can see in the left panel, and the right curve here is the Q-sum value of every single time point. As you can see, well, I didn't take I didn't take the absolute value for the red curve here. As you can see, if you go for the most extreme value, where the differences of the two parts of the sample mean have the most extreme values, that's actually the potential, the true change points. So that's the intuition of this well-established method, this Q-sum. And this is a one-dimensional time series. Remember our data set, we've already represented in a way it's a high-dimensional network time series. Okay, so this is a Q-sum, and of course this is one-dimensional, and recently there are many papers on a high-dimensional one, and also some of the papers also have already been used in the brain imaging community. Okay, I said there are two main components. One is change point detection in time series, and the other is network. And thanks to the previous two speakers, I can probably omit something here. So the second key component is the community detection. So for instance, here, this is a real data. This is the high school friendship data. So each node is a student, and if two students are connected by an edge, which means at least one of them claim they are friends. I omitted the direction here. If I say I'm your friend, no matter how you think, you are my friend. So this is the network. And using some methods called spectral clustering. Spectral clustering, we can think this is a convex relaxation to solve the stochastic block model. So if you are going to ask me about how many nodes I can handle, I can tell you a lot. So what else do I want to say? Anyway, I can talk more about details if you're interested in, but let me just proceed. So anyway, we have a spectral clustering methods to detect the community structure in our brain, in a network. Finally, we come to the method. This method goes in this way. Okay, so anyway, I can't find it well. Okay, so the data matrix is Y. The data matrix is Y, and it's a T by P matrix. T is the number of time points, P is the number of the nodes or the region of interest, voxels, or whatever, the dimension of network, the dimension of time series. I want to see whether or not a certain time point is a change point. I simply go through every single time point, say this delta J. I split it into two, okay? I split into two, this is what we see in the Q-sum in the binary segmentation. And then for each part, I do things parallelly. For each part, I first construct a correlation matrices. This is where the functional connectivity kicks in. Correlation matrices, and for this correlation matrices, I do a spectral clustering because I assume our brain has a community structure. Different parts are working collaboratively together. And this spectral clustering will give me the community labels of each node and the centroid, basically where it lies in RK space. It's just a dimension reduction representation. So I go to spectral clustering, and then I have the centralized matrices. So this is basically, I do dimension reduction from a P by P matrix, P is the number of nodes, to a K by K matrix, K is a pre-specified number. I assume there are K communities in our brain structure. So for each part, I have this representation, low dimensional representation. And remember, in Q-sum, if you want to detect the change point in terms of mean, it's so natural that you say, okay, I just take the differences of the sample mean. At most, and standardize it, right? But if I say I want to detect the change point in terms of the network structure, think of, it's not that straightforward, right? So the thing we come up with is the singular values of this matrix. I'll say a little bit more later. Anyway, so when we get this Q-sum-ish stuff, and we will test whether or not this point is significant. And if it is, okay, this is a potential change point, and I proceed until I reach a certain stopping rule. Okay, just some details. So I'm using the correlation matrices here as my adjacency matrix, but why not using precision matrix? The thing is, this is a high-dimensional stuff. Whatever high-dimensional stuff we do, we know that we need some sparsity. So what kind of matrices you are using basically depends on what kind of the sparsity assumptions you are assuming. If you want to go for the precision matrix estimation, that basically to say we know whatever good estimators requires the row sparsity, which means as for a certain voxel, certain region in our brain, at most, even though there are like thousands of other voxels, there are at most a few other voxels which are correlated with it, okay? That's the assumption you need if you want to go for the precision matrix. But we go for the sparsity assumption that although we have like thousands of voxels in our brain, but there are only a few communities. That's why we go for the adjacency matrix, which goes back to the correlation matrix. Okay, so that's one thing. Yeah, adjacency matrix using practice, the choice of K. I don't know if you saw the footnote of the high school friendship network. We do have a paper on how to choose the number of communities, because if you know a little bit of spectral clustering, you know that we need to choose the number of communities before we start doing everything. Although we do have a paper, I don't have a good answer here. Tuning parameter selection is always a problem. And always challenging and open. But I would just want to say, here, our method is robust as long as you are overestimating. So I think it's bad news and good news. The last thing is a more technical detail I just want to omit here. Okay, this is go for the singular values. I said if we want to detect the change in terms of the mean, we take the differences. That goes to the Q sum. So here we want to test the differences between two networks, and we've already used this lower dimension representation. So that goes to the distance between two subspaces. So what can we use? And this is the thing we use. It's called principal angles. The idea of principal angles is a generalized version of acute angles. So if I give you two unit length vectors, only two, I ask you the distance between these two. What can you do? You can take the acute angles between these two unit length vectors, right? So a generalized version, if you have multiple vectors instead of one each, that's the principal angles. That's what you should go for. And there are other ways. I don't know if I have time. Anyway, just show you some simulation results or numerical stuff. So this is a select. Thanks. This is a selection criterion. Basically, you remember I have this red curve, black curve, and as for the red curve, we go for the most extreme value. And here, because we are taking the singular values is actually the arc cosine of the principal angles. So the larger the angle is, the smaller your singular value is. And we just go for the smallest one. And we split until either we can't find the significant points, or we what else? Or there are too few points to proceed. And there are, I only have 5 minutes, but well, we can talk more in tomorrow's session about this, the more theoretical side. Simulation. So we have a few simulations. So I just go for the most left one. So you can see I have like two chunks, and basically the one in between is the change point, and the different grayscale is different community, and I just generate a network like that. And I have different settings, different P, different T. And this is, so basically the peak of, this is the density of the network changes, the change points we found, and the red vertical lines are the true change points. And there are some numerical results. I just want to show this one for a bit. So for the most left one again, it's a little bit hard to interpret, but the thing is, you see there's a change point, and that change point is estimated by our method. okay? And the above that line, above that timeline, the nodes are colored by the true labels, okay? So I just want to use this to show that, yes, this is indeed the change point, because after this, the colors mixed. We also have real data, and real data, this is also one of the motivations, the resting status, because if you know what kind of a task your subject is doing, you know when, for instance, he was sleeping and then he suddenly he starts singing, you know that's a change point, right? But if you just ask your participant to rest, not to think of anything, so what you can get is the resting state one, and you don't know where the change points are. So this is our real data. And we find different change points for different people. I don't know. Okay, one last thing. Remember what we did. We use these principal angles ideas. We use the community detection ideas to find the different change points in one participant, okay? In one person. And we can find the different states one goes through, okay? We can use exactly the same thing to compare different people, right? So after doing change point detection for every single individual, what we can do is we can again use the principal angle idea to see how similar different people are thinking in different segments of time. And the only trick is, instead of going for the smallest singular values, which corresponds to the largest principal angles, we go for the largest singular values, which corresponds to the smallest principal angles. That means these two people at that segments of time, they are thinking similar stuff. The good thing about the brain imaging real data is there's no ground truth. Okay, I should stop. Anyway, yeah, this can be definitely applied to other areas as long as you have high-dimensional network time series data. Thanks very much.